All right, good morning, everyone. I'm Ben Holmes. I am the co-founder and CEO of Nanochon. We are an early-stage medical device company which is developing a regenerative implant for treating some of the toughest challenges in joint health and preservation today. We are specifically targeting something that is referred to as a focal cartilage lesion. These are essentially potholes that develop in the cartilage surface of really any joint, but they are most prevalent in the knee. They can be caused by a traumatic injury, but they're really more of a significant problem because they are the moderate to severe stage of osteoarthritis. So I think everybody is probably familiar with the term osteoarthritis. Degenerative disease of the joint eventually leads to the formation of these lesions. And they're an issue for patients because they do cause pain and loss of function of the knee and loss of quality of life. But if they're not addressed at the point of diagnosis, they lead to the continued breakdown of other tissues in the joint, and this eventually takes patients down the path to needing a total joint replacement. So there is a real desire, especially in the sports medicine vertical, to treat focal cartilage lesions sooner, much closer to the point of diagnosis, but there is not currently a go-to solution which is both effective for patients and also economical and easy to use for surgeons. And so that's where we see Nanochon fitting in. We have a fully synthetic, low-cost platform. It's based on a polymer composite blend, which actually replicates the collagen bundles in cartilage. So the material has cartilage-matched mechanical properties, but it also acts as a really good substrate for stem cell growth. And then we're actually 3D printing that material into a more coherent, well-organized matrix. So for us, 3D printing is not about patient specificity, but it's a very efficient way to create this structure. And so what would happen is the surgeon would slip this device into the joint in a fully arthroscopic procedure. They would use it to fill in these lesions after they've been debrided of the damaged cartilage. And then immediately upon implantation, there is absorption of blood up from the subchondral bone, which also carries cell populations. Over a period of several months, the patients would experience full integration of this device with new tissue, so the defect effectively has been completely filled and stabilized mechanically and with new tissue. And then over a longer period of time, the device and the material degrades away while supporting that tissue that grew to form healthy natural hyaline cartilage. And so from the patient's perspective, this is something that can provide immediate relief, very fast return to full activity, and then provide real cartilage, which is the key to unlocking longevity and truly reversing the osteoarthritic state. In terms of what we're looking to disrupt right now in the clinic, there's really three main treatments. The first is microfracture, which many have heard of. It's a purely surgical technique which causes the lesion to fill in with a scar tissue. It does provide pain relief, typically for 2 years, but the issue is it does not halt the osteoarthritic changes to bone. And there actually was a clinical study published several years ago that shows that microfracture-treated patients actually end up being worse off than patients that were just not treated at all. In terms of the alternatives to microfracture, there's really two right now. The first is cadaveric allografting, which is what it sounds like. A piece of cartilage and bone are grafted into the lesion site from an organ donor. The major issue with this is there is some cost concern, but it's very hard to get this graft tissue. Patients wait on average 6 to 12 months just to receive that treatment. And so in response to that, there are some device and pharma companies that have developed cell therapeutic products where live cells are cultured or enriched or embedded in some sort of deliverable material. The big problem with these is they have not demonstrated better outcomes than cadaveric allograft, but they're up to 6 times as expensive. And so they're a real headache for clinics to use. There continue to be issues with reimbursement. And then they also have difficult workflows, either being too stiff or too soft to use arthroscopically. So they require lengthy open procedures, which hurt the outcomes for patients and also take valuable time away from the surgeon. So we are preclinical, but we have done a wealth of safety and efficacy testing to date. It's been in 3 animal models. This is a snapshot from our goat study where we treated a 1 centimeter lesion compared to microfracture and took it out to 4 months. This has sort of been the bedrock of all of our development to date. We showed healthy hyaline cartilage and not much happened in the microfracture group, which was not a surprise. But we also have a pretty substantial ongoing project in horses. So we did a 2-month pilot where we did a fully arthroscopic delivery of the device to ensure that it could accommodate that and retain its qualities. And the good news is after 2 months, as I mentioned, you see complete integration of the device with new tissue, growing into the device. You also, on histology, see the beginnings of the formation of healthy cartilage, and you see total preservation of the other healthy tissue in the joint, which can often be a challenge with these types of implantables. We also have continued to generate data from a cohort of horses which we're taking out to 8 months, which will be part of our FDA submission, continuing to confirm all of the great bioactivity and stability we've seen. I think a picture's worth 1,000 words. On the left here, you see standard of care, which is growing something that's sort of a mess, frankly. And on the right, you see Nanochon, fully integrated, fully stable. And I think very interesting as well are the quantitative scores. So there are a number of different systems that surgeons use to evaluate these types of repairs. I think the most impressive here is the ICRS score, which is a 12-point scale. Healthy cartilage is a 12. Nanochon at just 3 months scored an 11. So the results really have been nothing short of phenomenal. We've spent a lot of time evaluating the market and specifically talking to surgeons. We've probably spoken to close to 200 surgeons to date. And I think we really are onto something that's valuable for them, not only because this is going to be a better therapeutic for patients, but because we've really thought about usability and workflow. As I mentioned, this will be able to be used in a fully arthroscopic procedure, which really opens up the full range of cartilage surface corrective procedures in the knee. We're targeting a $2 billion market opportunity annually, and that's based on a patient population of close to 500,000 annually in the US. But we also believe that the technology is very adaptable and very scalable. And so if you think about transitioning to treating lesions in other joints, that's close to a $4 billion market opportunity annually. And then if you think about scaling the technology as a platform to treat soft tissue virtually anywhere in the body, we think that's close to a $10 billion opportunity annually. To talk briefly about myself and the team, as I mentioned, I'm the co-founder. I've been leading the company since 2016. I've taken this company through a number of very high-profile accelerators and incubators. That's really how we've built ourselves up as entrepreneurs and built the company from the ground up. I also want to highlight that we are currently a Johnson & Johnson J-Labs company, and we are a Quick-Fire awardee. My co-founder, Dr. Castro, is also a very highly decorated and experienced researcher in biomaterials, 3D printing, and cartilage regeneration. He's also participated in a human clinical pilot for a 3D printed orthopedic device. We also have a very strong team on the management and advisory side. There's probably close to 200 years of medical device experience up here on this board. We also have some of the big KOLs for industry and a lot of domain expertise. There's also multiple exits up on this slide as well. And we have very strong development partners which are helping us both develop the product and shepherd it through the various gates to get to commercialization. We are currently raising a Series A. We're targeting $8 million. This funding is primarily going to go to do a first-in-human clinical trial in Australia, and the funding will get us all the way through IDE submission to the FDA. We also want to continue to work with our development partners to finish validation and verification, get ready for eventual commercial launch, and then we want to continue to work with our partners both on the regulatory side and the IP side. Talking about IP for a little bit, we currently have 2 awarded patents and 4 active families in both the US and Europe. We also have plans to expand the portfolio as well. And then also on the regulatory side, we've worked both with an excellent in-house clinical consultant and MICRA to define a strategy to get through PMA approval. I also want to highlight that Nanochon got FDA breakthrough device designation in 2022, and we've already used that pathway to get valuable feedback from the FDA. So again, my name is Ben Holmes. We're Nanochon. If you're interested, I hope you find me after the talk and at the conference.